Hello and welcome to BioRules video lessons, higher level edition. Today we are going to talk about electron transport, which is also known as chemosmosis and oxidative phosphorylation, because it involves movement of stuff, of chemicals across a membrane, so that reminds the idea of osmosis, although it's more to diffusion, and oxidative phosphorylation because it uses redox energy, oxy-reduction, to do phosphorylation to produce ATP. So, for us to talk about electron transport, which is the end of the aerobic cell respiration, we have to start reminding ourselves about glycolysis and link and Krebs cycle, the beginning of the cell respiration. So in glycolysis, we get one glucose, we break it down, and in that process we produce two ATPs overall, we produce two NADHs, and we have pyruvate. Pyruvate then enters the mitochondria. One carbon dioxide is taken out, produce more one NADH per pyruvate, 2 per glucose, and we have acetyl coenzyme A. Acetyl coenzyme A goes into the Krebs cycle, and by turning the Krebs cycle, you produce more NADHs, you produce 3 NADHs per acetyl coenzyme A and 1 FADH2, which is a fancy NAD, twice as much if you get for every glucose. So in this process here, we make a lot of NADHs plus H2 and FADH2, which are electron transport full of excited electrons. So overall, we have 12 transports of electrons, lots and lots of electrons, and now all we have to do I mean mitochondria, is to put these excited electrons to move. So by making these excited electrons to move, we are going to be able to extract lots of energy that came from the glucose by splitting it apart. Okay? So for us to see the electron transport chain, we have to understand the mitochondria. So all this process that happened so far, glycolysis happened outside of the mitochondria, link and Krebs cycle happened in the matrix of the mitochondria, and the electron transport chain is going to be happening in the interface around the inner membrane. So basically we have matrix on the bottom, and we are going to be having the intermembrane space on the top. So this here is the inner membrane with a few proteins. Intermembrane space, so basically is the space between the outer membrane and the inner membrane. Very, very narrow, very small volume space. And in the bottom we have the matrix, the equivalent of the cytoplasm of the mitochondria. So how is this stuff here going to happen? Remember, NADH is going to come here, it's going to suffer oxidation. Oxidation is loss, reduction is gain, oil rig. So it's going to suffer oxidation, it's going to be giving electrons to this first protein. By giving electrons to this first protein, it moves around and it provides enough energy for this protein to pump protons. So this protein is a pump. It pumps protons, but instead of using ATP to pump protons, this one uses redox energy, uses the energy of moving electrons to be able to pump protons. So the electrons from this NAD, they move through this protein and they are going to carry on. And you have the FADH2 also giving electrons over there, and they are going to be going around to the next integral protein, and they will move around, and when they move around, they pump more protons, and then they keep moving and they go over there and they pump more protons. So basically, what are we doing here? We are transforming redox energy, electrons energy, into kinetic energy of protons. We are moving protons using electron energy that came from chemical energy into the glucose. However, electrons cannot just move and move and move. They have to go somewhere. So the ultimate acceptor of electrons is going to be oxygen. So half O2, which means one oxygen atom, is going to be getting the electrons that are moving over here. They love electrons. And they're going to put together with two protons. And when they do that, we, voila, make water. So basically, why do we need to ventilate and to get oxygen into our bodies every single second of our lives? To be able to have this. If we have oxygen, all this here happens, and we produce water by making the electrons to move. And when the electrons are moving, then we have protons being pumped. So this is why we need oxygen. I'll talk about not having oxygen in a while. Okay. Now, what is the idea here? If you have 10 electron transports, actually 12 with the Faraday-2, we are going to be pumping a lot of protons into that tiny little space called intermembrane space. So the kinetic movement of protons is now creating a potential energy of protons. We have a lot of protons in the intermembrane space, very little into the matrix. The few ones that are here, they even go there to make water. So now we have the potential to make these protons to try to escape desperately. And if there is a space, they will escape. And they escape through this molecule here that's called ATP synthase. They escape down by moving down this gradient, which is the osmosis part of the name here, the chemiosmosis, which is more for a diffusion. The protons move the ATP synthase, so creating kinetic energy in the ATP synthase. And this kinetic energy is used to glue ADP plus P, making ATP. So ultimately, this molecule here is a tiny little nanometric generator that transforms kinetic energy into chemical energy all over back here again. So ATP synthase, the smallest generator in the planet. So this here is the idea of chemiosmosis, electron transport, oxidative phosphorylation. So this here is what's going on in the membrane, the inner membrane, or around the inner membrane of the mitochondria. Here we have a clear look on what's going on. So NADH becomes NAD, and the electrons here pump the protons into the intermembrane space. Electrons move, and they pump more protons into the membrane space. Electrons move, they pump more protons in the intermembrane space. But they have to go somewhere. They go over here, oxygen, together with the protons, they make water, accepting those electrons and then letting all this oxidative process to go on. Lots of protons that have been generated here, they try to escape back through a gradient. And when they escape back here, they create a kinetic energy of ATP synthase. And this generator that's twisting around uses this kinetic energy to glue ADP plus P into ATP. This is the process of oxidative phosphorylation. Okay? If it went too fast, go back in the video, watch it again. This is going to be important for you to understand what is going on. Okay? Let's take a look on this tiny little generator, one of the most wonderful molecules ever. Now, this is a protein, and as you can see here, a quaternary structure, many subunits, and they are assembled in a way that you have moving parts that when they move, so protons come in here, they end up moving and creates kinetic energy, and this kinetic energy is now capable of getting this guy here, ATP plus P, and becoming ATP. So it is actually a generator. Now, in human engineering, we use generators that usually transforms kinetic energy into electricity. In this case here, we are using proton potential energy to move protons, so proton kinetic energy, to be able to twist this here kinetic energy of the generator, ATP synthase, to be able to produce ATP. Amazing molecule, no, fantastic, one of the best ever. Putting everything together, let's see what goes on during cell respiration. We have a glucose that in glycolysis is transformed into pyruvic acid or pyruvate. In that process, we make 2 ATPs and we make 2 NADHs. Okay, 2 NADHs over there. Then the pyruvate goes into the mitochondria in the matrix, and through the acetyl coenzyme A reaction or link reaction, CO2, 2 CO2s out of the 6 that you had originally in the glucose are put out, and that gives 2 more NADHs, electron transports, full of electrons. And then the acetyl go to the citric acid cycle, and 4 more CO2, so at this moment the glucose is gone. Now they are produced, 2 ATPs is produced as well, and we have a lot of NADHs and FADH2s produced in that step. So all that will go to the electron transport chain around the inner membrane of the mitochondria. If we have oxygen to make water, then the electron transport goes on and we make a lot of ATP. So in the entire process, we will make about 38 ATPs. But what if we don't have oxygen? So let's have this here. So there. If we don't have oxygen, so no oxygen over there. If we have no oxygen, what's going to be happening in here? No oxygen, no electron transport chain. So we stop making those 34 ATPs. Actually, without that, we also make, let me put this over there. Okay, so without oxygen, this here stops. So that means we don't make those over there. So without this, also the citric acid cycle stops. So we make none of those 3 ATPs. So we also don't make those NADs. If this stops, this also stops, and we make no NADs in here. So in the end, what are going to be left if we have no oxygen? We are going to be having 2 pyruvates produced that cannot be used, and they have to use those NADs to be transformed into lactate, or in the lactic fermentation, or in ethanol and CO2 in the alcoholic fermentation, and the overall production is 2 ATPs. So without oxygen, 2 ATPs instead of 38, and that's ridiculous. That's very, 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 very little. However, if we do have oxygen, so oxygen here to make water, then all this here can go on, because with oxygen, then you can have all those steps going on. We produce 34 ATPs because all this here, they can keep moving, and we have a lot of ATP. We will end up having 38 with oxygen instead of having only 2 ATPs. This is the difference between aerobic and anaerobic respiration. Aerobic over here, anaerobic over there. So I hope you have enjoyed this class. Oh, I wanted to make it prettier, but okay. And I'll see you in another class. Now do the quiz and bring your doubts for us to discuss. If you didn't get much of this, I strongly advise you watch again the video. Okay? So you can come to the class with fresh doubts for a good, decent discussion. See you. Bye-bye.